field, the subfield to very closely watch for everyone is agentic coding. I think the role of this agentic approach of LLMs in speeding up software engineering is a very, I think, will unlock or will hold the answer to a lot of the other questions, including science. Science is harder than programming in some ways, but I'm seeing, I think, the same paradigms, the same technologies, and same, I don't know, agentic workflows and so on that are going to work for creating complex software will can probably be adopted at least as a first draft for co-scientists.
Hi, everyone. Welcome to episode 19 of the Pollock Principle. And I'm here today with Shantanu Singh from the Broad Institute. It's a very, very prestigious university institute. And I think we're going to have a very interesting conversation about, I don't know, like cells, phenotypic assays, novel ways of running a research lab. We have all sorts of really interesting ideas in our list of questions. But to begin with, I'd love to hear a little bit more about your path, your story, like what brought you to, research and how you ended up at the Broad Institute, and what kinds of things are interesting to you, please.
Sure. Thank you for having me, Javier. It's a real pleasure to be on this podcast. We just chatted before this and mentioned that I really enjoyed the conversations you've been having with folks in our field. It's such an exciting time to be doing all this. I'm so jealous of people who are doing their PhD right now because everything seems to be up for grabs right now in terms of solving it. But yeah, so about myself. So I'm Shantanu Singh. I'm a group leader at the Broad Institute. I'm the co-PI of the Carpenter Singh Lab at the Broad. We are a group of computer scientists, machine learning, data scientist folks who are focused on this idea of using computers to look at images of cells and then extract information from them in a way that can then be used to somehow characterize their cell state. And we'll talk about that a little bit, characterize the disease state, and then potentially find drugs that may cure the disease. We'll talk about more about the research, but you asked about my background. I'm a computer scientist by training. Started my undergrad, my PhD, all of that in computer science. I was a computer geek as a kid. Used to go about, doing things like installing Linux on my friends' computers and try to convince them that that's the right thing to do. I still think that is true, although I'm using a Mac right now. And then I was very fascinated with this idea of AI even back in the day. This was during one of the many AI winters, and one of the things that really fascinated me, the path to AI that fascinated me, was that of computer vision. I really thought, if we can get computers to see, then that might be the first thing. That perceptual pathway into intelligence might be a great way to explore this further. And I did some of that during my undergrad. I joined Daimler Chrysler, now called Mercedes-Benz Research in Bangalore, to work with them on a bunch of things. That was sort of a very open-ended lab at the time. And one of the things that they were doing was trying to figure out if they can predict the behavior of pedestrians crossing the road. So the early C-class models had these cameras mounted on top of in front of the cars, and there are all these pedestrian data sets that that we captured that would then allow us to then train models that could then identify if a pedestrian is crossing the road. So this is very early stages of self-driving cars, and I sort of that was my early foray into applied computer vision. Then I went on to do my PhD in machine learning and vision, focused initially on this video surveillance problem, which was both very difficult and also a little spooky. And I sort of slowly moved away from that and started thinking about how I can apply this to problems that are, for me, more motivating. And I found that biology was an excellent domain to which I could apply this. I found that to be, I thought initially it would be easy. And like with most things, it starts off as, something that you could probably do in a day, and then it takes a week to get, 50% of the solution, and then the rest of it takes your PhD. And in my case, it was this problem of identifying structures in tissues, in breast cancer tissues of mice, and trying to identify structures in them. I built these 3D statistical models of cells, and I imaged all these tissues myself under a confocal microscope. It was pretty crazy. But then I could still do, maybe 10s, 20s, maybe, I think I collected maybe 200 cells in terms of their very detailed 3D shapes and things like that. But to really scale it up, like if you really want to be able to do anything substantial in terms of being able to predict stuff or, be able to do anything that can be eventually used to understand biology, you really have to do it at scale. And the Broad Institute is known for that. Sometimes notoriously to the fact that they scale up everything. And in this case, scaling up imaging was such a tempting thing to do, because if you could do this in high throughput, then you could really generate the kind of data you need to build predictive models. And I didn't really know too much about this field of high throughput imaging at the time. And I was sort of, someone like a siloed computer scientist at the time. And then, I learned about this whole field. And Carpenter at the time was leading this lab on actually developing Cell Profiler, which was this bioimage analysis tool that's now widely used, and I think a predominant tool for any high throughput imaging. And she was slowly starting to think about this idea that you can now actually take pictures of cells at scale and use that as sort of rich information to characterize cell state. So I joined the Broad really with this greed of scaling up everything I was doing 1000x, and I eventually, long story, had stayed on to create a lab, and now I co-lead the same lab with Ann Carpenter. So that's my somewhat. If you ask me ahead of time what will happen next, I had no clue what would happen next. So as life goes, one thing leads to another, and I'm here right now. We have a lovely group of folks who are just motivated with this idea that you can take pictures of cells and reveal so much about their biology.
Yeah. Let me try to go a little bit deeper into that. And of course, I'm not a biologist, right? So please correct me if I'm wrong. But I think so. Now, maybe it seems obvious, right? But this idea of using images to do high throughput biological work is fairly novel, right? Back in the if you go back, I don't know, a few decades, right? If you wanted to do an assay on a system, right? When you go to the doctor and then they check your blood glucose or whatever, you take the sample, you do your assay, you get one value, right? You want to do, 100 of those, and you do one at a time, and then, it takes, forever, right? If it takes, a minute to do one, it takes 100 minutes to do 100, right? And then when people started doing genome sequencing, there was this really interesting idea. And I think it was the Illumina machines, right? The ones that started doing that, where, you do all these chemical reactions that trigger light, and then the cameras capture it, right? And then, you can kind of, get, by recording, those images, you can do, a million, different, analysis at a time, which then, you kind of, put together into a genome, right? And then this notion that, you could do the same thing with cells, right? This is, novel and imaginative, right? Because before you had to get your sample, put it in a microscope, poke it, I don't know, manipulate it, I guess, with some kind of, tool, right? And then record it one at a time. And then, somebody comes and says, well, why don't we do, 100 or 1,000 or 1,000,000 of them, and then just record it, and then using this, massively parallel technology of a camera, right? That is the ubiquitous, right?
Yeah.
So I think that was, again, now it seems obvious, right? But I guess in retrospect, this would have been a, revolutionary, change in how people do things, right?
Yes, I absolutely, and I couldn't phrase that trajectory better. I think the perspective I would share here is that there are probably three aspects to it that resulted in this massive scale up, right? One is that you already mentioned, the fact that you could just take pictures of cells and they have rich information in them, and you can read out not one thing but hundreds of things, and we'll come back to that. But then there's also this other thing of robotics and of high throughput imagers that are just able to record things at scale very, very fast. And the field of high throughput imaging, where you can basically take these, for the listeners who don't know what multi-well plates are, these are essentially, you can just think of them as probably the size of a large iPhone or something like that, a small tray with really small test tubes. And in each test tube, you can do a separate experiment. These test tubes are called wells. And so you can do all these experiments, and these are typically the workhorse of high throughput anything, high throughput imaging included. And that technology of just being able to do experiments in high throughput, that sort of had started picking up pace. And then separately, from the point of view of perturbations, we've always had large chemical collections, I guess. But you know this better than I do, Javier. Now it's possible to do that first in silico, and then actually go and produce the chemicals that you care about. I don't know too much about that field. as much as you do. But there's also this genetic perturbation aspect, right? It became possible with CRISPR, but also preceding CRISPR ways, the high throughput ways of really scaling up that. So all these things started coming together, which is why it's such a wonderful time to be doing all this research. But the part that I'm more, I guess, of an expert at is amongst those three things is really the imaging part, the first thing that you talked about, and that has a very interesting trajectory because we've been microscopes were among the was probably the first technology that we humans used to start studying biology in detail, right? 400 years ago. And this idea that you can then take, pictures of it and understand something about the biology based on this appearance, that's been around for a couple of hundred years. But the going from there to then taking digital pictures of it and understanding what's in that digital picture, that took a little longer. Obviously, maybe the last 30 years we started taking pictures and then using image analysis techniques. No fancy computer vision and image understanding, anything. Just identify the cell, use standard algorithms, watershed or whatever, to segment the cells, extract standard features, classical features like texture variations and things like that, to then characterize a cell. That started becoming a thing, but still, I think biology was still, and I'd written this paper in 2014 as a postdoc about increasing the content of high content screening, which is that you can take all these beautiful pictures, but it seems like what the field is really doing right now is taking one or two measurements from it and going with that. Like biologists really care about they care deeply about understanding what they're measuring, I think. And one can debate that, but I think deeply this idea that, I want to measure this one thing or these two things, and I'm going to take all that. That's all I'm going to take from those images and go with it. I think that at least that stuck on for a bit. I mean, people would take all these images and measure these one or two things and make a lot of discoveries. And even that was hard because getting those measurements reliably is hard. Okay. But then going from there to saying that, okay, I don't want to just directly measure one or two things like the intensity of a protein or the size of a cell. I want to capture more complex phenotypes. For example, a metastatic cell or a migratory cell is something that you can't directly measure. Like you have to train a classifier or something like that, right? Yeah. So then these methods started coming up. Cell Profiler Analyst, one of the tools developed by the lab that I now co-lead, and a few others tools are out there that essentially were ways of just sorting cells into bins and then training a random forest model or something like that to identify phenotypes, right? So now you extract a whole bunch of features, but then you still predict one thing, which is this phenotype present or not? And what is your degree of confidence that it's present? Yeah. And that, I would say, that held on for, that still is a very, very important path to doing all of this. But that was kind of the state of the art until around the time when I started digging around in the field. And then this idea came that, what? We very often don't know what you're looking for, but you kind of believe that images contain rich information that can tell you broadly about what's happening, exactly like those who know transcriptomics and all that. The reason you read out everything in the gene partly is one, because you can, and the other is because you really don't sometimes know what you're looking for, right? And if you can get a broad fingerprint from these images, then that broad fingerprint can then be used downstream to find things that you care about, right? So this field is called image-based profiling, and that is a very different take on how to use images. Strangely enough, for folks like me who came in from computer vision and computer science, it seemed like an obvious thing to do. I don't say this in a haughty way. It's just like literally, I felt, okay, yeah, isn't this the only way you should look at images, which is like featureize them and then use it for whatever, like cluster it or whatever. But it was not so obvious, and it took a while for us, for Anne and me and others in the field, to kind of convince biologists, convince our colleagues that images and used in this way are as informative as transcriptional profiling or proteomic profiling, and they are cheaper. And we wrote a few papers on showing how the same problem you want to solve, like you want to predict, let's say, and maybe we'll talk a little bit more about this in detail, but just broadly, you want to predict the mechanism of action for compound based on the transcriptional profiles using a guilt by association. We said, okay, let's do exactly the same thing. You use mRNA profiling, we'll use cell painting. We'll do this experiment together in a paired way. And let's just see how we do on a downstream task. And it turned out that it did as well, if not better, and it was cheaper. So anyway, so that is, a very, long history of this field, but it really has a lot of detail and texture to it as coming up to the point that we got here.
Yep. And so, maybe to understand a little bit better, like the
Yeah.
kinds of things that you can do or cannot do, right? Like, so with, um, I guess, optical microscopes, there's a limit to how much resolution you can get, right? Like,
Yeah.
what kinds of things can you see in a cell? Can you see, I guess, like the outside shape of the cell? Can you see inside? Can you see the nucleus moving or like the nucleus, present inside of the cell, like if the cell's transparent or not? Like, I mean, like for people that I guess it's kind of, hard to explain in a podcast. Some people may be just listening to it instead of watching the video, right? But what are the kinds of things or the kind of phenotypic patterns that you can actually see with one of these instruments when you
Yeah.
take a photo of that?
Great. Yeah, I'd love to share a couple of links that your listeners can click on and just look at these pictures, which are worth 1,000 words, as they say. But I would say that microscopy is an amazingly diverse field, and I'm not a microscopist myself, and I don't actually know the full span of everything you can do with microscopes. The part that I really focus on, or rather the part that our lab tends to focus on in terms of the source of most of the data that we care about, is fluorescence microscopy done in 2D mostly. And with that, you can still do a lot, which is that so fluorescence microscopy, for those who don't know, is just basically that you take cells and then typically you do one of two things. One is that they might have this what they call endogenous expression. That is, you've somehow mucked around with the cells so that they are naturally able to express flow of proteins that glow. GFP, some may have heard, is one of them. And then you shoot lasers at a certain frequency, and then they glow, right? And then the other way you can do it is that you just take regular cells and then you go and stain them using some markers that go and stick onto some certain structures, and those structures then glow when you shoot lasers on them. Okay, so what are the kinds of things we can see? All right, so just sort of tying it back to this idea that I just told you about of like using images as signatures. One way, I mean, the idea is that you still have to now go and pick specific ways of imaging. Like they're specific. Like you're not, when you're looking at images in that broad way, you're not looking for a specific pathway, or you're not trying to study a specific pathway or a specific protein. You want to sort of get a broad state of the cell. And for that, what we did was that we tried to pack in as many dyes, as many fluorescent dyes, these colors, as we could fit into a single imaging assay. And what we ended up with is something called the cell painting assay. Some may have heard of this. Those who follow tech bio may have heard of this company Recursion that is sort of routinely has for a long time been doing this. And so cell painting was really their workhorse, and was a technology developed by our lab. And that idea was basically just that you can essentially mark things like, to get to your question now, things like like mitochondria, endoplasmic reticulum, ER, actin Golgi, plasma membrane, nucleoli, and things like that. And so the cell painting assay specifically can mark, I think they've counted about eight organelles that we image in 5 imaging channels using 6 stains. And that alone can give us a lot of information about cell state. Now, there's much more obviously you can do. One may often wonder, what about, I don't know, sarcomeres or something like that, which when you're studying heart cells, what about all the structures that are there, neurons? Like, how are you going to mark that? And the idea there is that you can still get a lot just with a few stains, but very often, our colleagues have adapted the cell painting assay to mark more specific things that the biology cares about. But broadly put, I'll share the link. You can just go and look at the images of cell painting, and you stare at them, and you say, "Oh, that's it! Like just these 5 channels, and that's giving you all this information." And really, once you start digging in and measuring specific things using computer vision techniques from them, then you can get thousands of measurements out that are correlated, but they're still, deeply informative of cell state.
Mhm. Interesting. Okay, so like let me try to see if I can apply it to like a real world thing that I may want to do, right? So like I think I think as I mentioned before, like I have an interest in longevity, for example, and some type of diseases and whatnot. So let's imagine that I'm interested in mitochondrial health, right? And I have this hypothesis that, that when mitochondria start, fusing, in a cell, that's a bad thing, for whatever disease and whatever longevity condition I'm interested in. So I want to come up with a molecule that promotes mitochondrial growth and fusion, right? And so I would go and say, okay, I have no idea how to, which molecules are going to better work. So I have like 1,000 different molecules that I want to explore, right? So I would go and say, that, fits into like, I don't know, 12 plates or whatever, right? So I get, my cells, and I get my, 1,000 molecules that I want to explore. And then I would use this technique with a stain that somehow binds to the mitochondria. And then, once I put that stain, I can see maybe one image, right? Or like, how many mitochondria are in each one of those cells. But what if I want to see over time, for example? So I want to see, what happens, after I, perturb all these cells, over the course of, you know, a week, for example, right? Like, would that be the kind of experiment that I would do, in order to analyze that, or like, what is a mistake in kind of my experimental design?
No, no, no. I think that's a great starting point. So you have in this case, it's a little bit more like the second mode of imaging that I told you about, which is that you already kind of know roughly what you're looking for, which is that you're specifically focused on mitochondrial health, but not completely because you still don't know what is the thing that you need to measure to capture health, right?
Yeah.
I think the design would be pretty much that you would then start saying that okay, I will. In any good experiment, you'd want to have some positive and negative controls, and so now you'll start designing an image assay. By that I mean you'd have already designed, figured out how to pick the right set of stains for marking mitochondria, and then you will go and extract a whole bunch of measurements from the mitochondrial channel and then try to just do at the very beginning just something very very simple, which is that you would, I mean, simple depends on how you think about simple, but the simple for me would just be that you would extract unbiased measurements from it. You could take the so-called cell profiler measurements, this classical hand engineered features, or you could take, I don't know, the latest foundation model for vision, Dino V3 or something, and extract a high dimensional embedding of the images comprising just those mitochondria and go and cluster them and just see, okay, of all these 1,000 compounds I have, is there any natural clustering that falls out in terms of the way they are, in terms of the way that they're perturbing just the mitochondrial structures? Okay. The next thing you do is, I would do at least, is that I would then look at where do my positive controls lie? Like, am I able to capture do they lie in a particular part of this phenotypic space? And if it's very clear that, there's one place where the good thing that you want is happening, then maybe you're in luck and maybe you don't need anything more sophisticated. You can literally just go and screen compounds that seem to induce that phenotype. But there's still an extra layer that you have to now do, and which is why things like cell painting are important, which is that you might be doing very good on that task of, I don't know, rescuing mitochondrial health or whatever, right? You may have figured out an assay that specifically measures how well you're able to rescue that phenotype, you're able to improve that thing, that trait. But the compound could be doing a whole lot of other things you don't want, right? It could be like really screwing around with your cell. And figure that out, you really have to get a broader readout. And for example, something like cell painting can help you do that. Cell painting or anything that marks other organelles too. So you might discover that, when you're looking through the narrow lens, oh, my mitochondrial health is improving. But then suddenly you look at the cell as a whole. You look at, I don't know, suddenly there's a lot of ER stress or something like that. And you may not have realized it had you not also imaged the ER channel. So now what you do is that you say, okay, I've figured that out. I know what I want, but I also want to figure out that it's not doing things that I don't want. So you now go and get the whole cell painting profile. And you can do many different ways. One thing you can do is that you can just say, hey, does my cell painting profile as a whole of these compounds look similar to, I don't know, these promiscuous compounds that I've already collected that just hit a whole bunch of targets and do awful things? Are they similar to these toxic compounds that I know have been, I don't know, liver toxic in the past? And I don't want that, right? So that guilt by association kind of thing can get you very far. And then you can sort of iterate on that and finesse it.
And then when you do this, does that um, perturb the cell as well in a way that would, I imagine like it would change a little bit my experiment, like, I don't know, depending on like, if you add lots of dyes, lots of, fluorescent proteins, floating around, or like, how much does that actually messes up the cell itself, right? Like, so like, if I wanted to say, well, I imagine like, keeping this, cells alive for, a week, right? Depending on the type of the cell, it may not be that easy either, right? But like, if I start adding all these different dyes and manipulating these things, does that actually, kill the cell? Does it disturb it? Does it, make changes? Or like, how much can I actually trust that I'm not the one that is, messing up,
Absolutely.
causing that the changes, right?
Yeah. How much is the measurements changing the state? Yeah. I It's a great question, and I think that is exactly why there's a lot of excitement about this field that is now broadly called label-free imaging, which is that can you just not do any labeling at all, not endogenous expression, which we talked about, where the cells naturally express a protein. You might think that is not bad, but that is actually bad because it's doing something very unnatural. I mean, it could be bad. Stains, as you said, might also be interfering. Uh, so there are two ways to think about it. The first way is that it is true it is messing up, maybe. Um, but that is part of the experiment. And as long as that is one of the many unnatural things that we're doing in this whole experiment. We're growing cells in a dish on a plastic surface. We are, I don't know, these are immortalized cells. These are not the cells we actually care about. There's some other cell. There's a lot of unnatural things, and this is one of the many. And you can say, you know what? Whatever.
Yeah.
We'll figure it out downstream. We're anyway going to do something more follow.
On what? Right, like that's kind of where I was going to. Like if you use some transcriptome analysis, you have to break down the cell, break down the material.
Yeah,
like,
there you go.
Yeah, so it kills it. There's no way around this. So like I guess it's a little bit better, but I guess it's not perfect.
Not perfect, but yeah. So that's one, like, shrugging. There's a shrug emoji. I put that here. Whatever. But the other way you could do it is that just say that, you know what, we don't want to do any of this. We just want to rely on images that we can take without labeling the cells in any way at all. So that field is called label-free imaging. And your former colleagues at Google, I'm remiss to have forgotten the names, but Mike Ando was one of them. Google Accelerated Sciences and others, I believe, worked on this idea that you could essentially, I think the 2017 paper, that you could essentially take bright field image stacks, and so essentially no staining at all, just take bright field image stacks and essentially just, these images look very boring when you look at them. They look gray and you say, what the hell, where did all the color go? And then you say that, okay, I'm going to stain these same images with these fluorescent dyes, and I'm going to just try to predict those stains using these bright field stacks. In other words, we're trying to predict whether we're trying to test, but also predict at the same time whether just these raw sort of unstained images can predict actually the stained structures. And it turns out that you can go very far with that. There's a lot of information in those intricate patterns that don't seem obvious to us, and it is not surprising that we can't see those patterns. Our visual system was not trained to detect, to look at, to look at cells for that matter. We were trained to, like, spot a lion hiding behind a bush so that it doesn't kill us, right? So these bright field images are now, bright field being one of the many other ways in which you can image a cell without labeling it, contains a lot of information. And companies like Recursion, they've actually moved away from what I understand. They have at least reduced their reliance or their dependence on cell painting, on fluorescent staining, and instead they have this, I'm guessing, a very, like, well orchestrated, well built model that only requires you to take bright field images, no staining at all, and get the same information out. So that's a long answer to your question that yes, it might be doing bad things, number one. Number two, it might be unnecessary to do it if you can already do it using a label-free technology. And that is a very exciting field that's been around for about 7 years now, since Google and folks at Allen Institute first came out with the paper.
Oh, very cool. Yeah. I mean, Allen Institute is here also in Seattle, like a stone throw away from.
Oh, there you go. Yeah, that's right.
I was there actually once, back when I was still at Google, right? Like, we were talking with them about the cloud infrastructure. So I got a little bit of a tour of the lab. So that was nice. I had to put on a white coat and all of that. So one question that's coming up as you were talking about, right? Like, so we're talking about training classifiers or training models to analyze this data, but maybe a more interesting question is, of course, when you have this high throughput method, you get lots and lots and lots of data, right? Like, would you, how are you using that, to train your own, for example, foundation models for, self-preservation, for example, right? Like if you've had, a million, of these different, experiments, could you feed that into a model and then train a foundation model that says, well, if you do this to this cell, something happens to it, phenotype. If you do something else, the cell, loses its shape or whatever. And then, with that, I guess a second part of that is like, would that scale up to other things, right? Because I know like people have done this for transcriptomic models, for example, right? Try to create these models. So could you do that for, imaging, phenotypic, kind of models? Is there enough data to do that?
Yeah. No, that's a fascinating question to explore. And I have some thoughts on it. So the short answer is yes, we have trained our own models. There's one called the Cell Painting CNN, real version 1 model that's out there, developed by a former postdoc in our lab, Juan Caicedo, who now leads his own lab in University of Wisconsin. And his lab is really pioneering this in terms of building these foundation models for imaging. There are many others, but that's our direct hook into that aspect of it. But I just did want to comment on foundation models in general. It's a bit of a hot take, which is that just broadly for background for your listeners, foundation models in biology, they're these large AI models that are pre-trained on massive biological data sets, like you said, millions of gene expression profiles that can theoretically be adapted to many downstream tasks, like GPDs for cells. And the promise has always been that training it on huge amounts of biological data would create models that understand fundamental cell behavior. Okay. But I think my view is that the reality check really came earlier this year, maybe late last year, when studies showed that these sophisticated models were being outperformed by simple baseline methods, right? Sometimes even just looking at the 2,000 most variable genes could work better than models that were trained on millions of data points. So for me, at least, it was a classic case where, complexity did not necessarily translate to better performance. The jury is still out on that, I would say. I mean, I've not been closely following it, but the jury's still out. I know it's getting better. I know that there are specific use cases where it's tremendously better. But it gave me pause. I mean, I was I've always been very bullish on this idea that you can sort of the idea of representation learning. One of my favorite workshops is LMRL, learning meaningful representations of life at NeurIPS and other places. And I heavily lean into that. And I do think that that is the future. It's just that I have been surprised how difficult it has been, at least in imaging, to replace classical features that have been around for 30 years, where you can just extract predetermined features from images and use that for these downstream tasks. Now, having said that, there's emerging evidence that things like Dino V2, for example, this is the model by Meta, by Facebook Fair, and we haven't tested a Dino V3 yet, but off the shelf, like literally not even fine-tuned for images, off the shelf, fine-tuned for whatever, for natural images, cats and dogs and whatever. Use that and just plug it in and extract those features from single cells. And in 2 of our experiments, they were performing as well as handcrafted features, not better, but as well as handcrafted features. But this was without any pre-training. Like we did the laziest thing possible: Hugging Face, download, extract. That's it, and it worked. Now, can you improve on that? I certainly think we can, but we have to be very, very, very cautious to make sure we are sort of creating very good, strong baselines and benchmarks to truly evaluate what is the improvement that we can get over baselines, especially very lazy baselines like download from Hugging Face.
Yeah, yeah. I mean, it's sometimes kind of hard to figure out, what is the benchmark or what is the level of performance that you could practically or theoretically achieve on a specific task, right? Like, I think maybe a lot of people would know, like, in image image recognition, for example, it was only after what was ImageNet, Feli's, team, kind of put together this really large data set when folks were like, okay, now we can train on it. And then, we had this, massive achievement of like using a super simple neural net instead of like the much, much more complicated, models before, right? And like, I mean, personally, I don't have a good intuition of like, is that enough omics data today, right, to build a foundation model that really predicts whether a cell will respond one way or another. You perturb it with a molecule or with with some RNA or whatever, right? And somebody could say, yeah, we have enough data, we just haven't figured it out yet. Or or somebody could say, no, we need like 3 orders of magnitude more data in order to truly build something that captures the complexity of the cell. And I would I would have no idea how to pick one or another. And I've been kind of of course, again, I'm not a biology, but I've been doing this for already for like a year and a half, right? And I have no intuition, of in which of those two cases we are now.
Yeah, yeah. No, I'm with you. I don't think we are there yet in terms of image scale. If you look at it, so for example, um, I don't think we're there yet in terms of the amount of data we need, to be honest. And I wonder if there's some fundamental difference in the data types really compared to natural images. Okay, so I can sort of more narrowly address the data problem or the data situation from the imaging perspective. So we are a relatively small lab. And our ability to produce massive amount of data is limited. But what we realized is that to really scale up uh, data in this field for exactly the reasons you mentioned, but also just to make discoveries. The way for our paths to that was to create a community in pharma. So we created the JUMP Cell Painting Consortium in 2019. It was a consortium of 10 pharma companies and several academic labs, all led by us here at the Broad. And our goal was to create together the world's largest public cell painting data set. So we got together and we said, hey, we're going to we're going to agree on a set of compounds and genetic perturbations that we're all going to profile together. So we selected about 120,000 compounds and about 3/4 the human genome. And every lab then went and imaged a certain fraction of it. And we did that in 5 replicates. So every compound is actually replicated in 5 different labs. And the jump cell painting data set now has been publicly available since November 2022, and it's been, there's been a massive uptake, and people have been building models on that. That comprises 1.6 billion single cells, which is a lot of data. I've even lost track of how many images there are, but like individual cells, there are 1.6 billion profiles out there. The data is hosted on AWS. I take every opportunity to thank them because it would have otherwise cost us several tens of thousands of dollars every month just to host that data, and especially all the egress charges. And so it is a lot of data. People are using it. But I feel that in terms of the variety, like we have 116,000 chemicals now in that, and 40, whatever, and 3/4 of the genome. But it's one cell type. It is one cell type right now. We had to pick on one modality in which we could, in one dimension in which we could push. We pushed in the perturbation modality. And we kind of realize that now if I want to translate to a new cell type, we still need to run another experiment, not at the scale as we did here, but I don't know, maybe for a small subset at least, so that we can solve this translation problem. We don't even know really if that translation problem can truly be solved. But we are at a point where, at least for this one cell type, for the U2OS cell line, osteosarcoma cell line, I think we have probably for that we have enough data. I don't think we need too much more data. We have 1.6 billion cells, a lot of perturbations, but we have a lot of different cell types. We have a lot of different imaging conditions you can consider.
A couple of cell types and human, stuff like that.
Yeah, the concept of a cell type itself is under debate as to what is a cell type. There's some debate whether it's actually a discrete thing, but I would say if someone threw out the number of 200, that would be reasonably acceptable. Um, so, and a lot of them happen to be immune cells, I think. So it is, there's a lot to profile there. I don't think we're there. I don't think the answer will be to do a dense sampling of it. I think that is impossible. So some way of sparsely sampling the space of that massive hypercube of cell types and perturbations and technologies would be is probably the next frontier.
Some cell types would just not necessarily grow the way that they would normally do inside of a petri dish, right, or inside of a of a tray or whatever, right? And some cells are easier than others, right, to grow that way. And Yeah, I guess that's why, one of the reasons why like there's this push now towards organelles, right? I mean, like on the discovery side, I think people have realized, well, if we have like organelles instead of like cell samples, it's just a little bit better data on the drug discovery side, right? Like the problem, of course, that is more expensive, and we still don't have a lot of the technology to to develop them. But I think people figured out how to make like a good dozen or so different cell types into organelles for like liver and so on and so forth, right? That is a little bit gives better data, right? As though like you know you're perturbing it in the way that you want. Um and one of the things that you mentioned also at the beginning was folks inside of your lab. If somebody was doing a PhD, or like I guess for the folks that are doing PhDs in your lab now, what are the challenging problems? What are the things that that are still, or that are maybe more top of mind, right? For folks to discover or to like figure out.
Yeah. Oh my God. Yeah. I think you're going to get me excited. I'm doing a PhD myself right now because there's just so many things I want to tackle immediately. But the one thing that has been that has been bothering me for a while, since 2012, I would say, has been the idea that I told you about the fact that we can get from a single well, we can get something like 1,000 cells imaged. And we have collected 1.6 billion cells from the jump cell dating data set and things like that. But it turns out, though, that when you're actually translating those profiles, we have extracted these features from cells. We have now taken all those cells, and we are now using that collection of cells and the high dimensional embedding of the cells to represent cell state, right? Now, how does that set of embeddings, how does a set of features represent cell state? The simplest way you can do it is to just aggregate it. You say, I'm going to take the average cell for the sample. I'm going to take the average cell, and that average cell, that average profile, which is literally just the mean, is now going to be the embedding the feature vector that will represent that sample. So, for example, every chemical I have now that I have profiled maps to a, I don't know, 1,000-dimensional feature vector that is the average of all the single cells that I collected for that chemical. Now, that would be the first thing you'd do. And you'd think this is the simplest baseline ever, right? Averaging all the cells. Turns out doing anything better than that has been extremely hard. It's been extremely hard to get better than just computing a mean. You would think that there's all that rich single cell data that's available, and you can surely get higher dimensional information, I mean, higher order information from it. And we've tried, I would say, 3 times, 3 different ways. Each of them have gotten us a little bit of lift. We have figured out, there's this deep sets approach where we said that we can try to get a embedding of this by learning the set representation. We tried out some higher order statistics, and we tried a few other things, clustering based, and none of it was convincing enough, I would say, for us to change our ways completely. That is, let us do anything better than computing the averages. And that is ridiculous to me. Like you look at these images of cells and they look so diverse. Like even even a very strong perturbation, there's a lot of variation in them. And it has to be the case that that variation, that natural heterogeneity or the heterogeneity that's induced by perturbations, that heterogeneity, the variance, the standard deviation, if you think about it, but in a higher dimension, that has to be informative, right? But it doesn't seem to be something that we've been able to capture. So I think someone who can develop methods, and there are a few labs who are actually working on this. We're collaborating with them on that, on developing. They're using different approaches. There's this optical transport-based approach. There is a learning-based approach that is just uses sort of a bunch of dictionaries. They're trying out different ways of doing it. But if you can solve the problem well, I think you'll unlock a lot of information that I think is hidden away. That's number one. Number two is actually a seemingly boring problem of batch correction, and I'm sure, Javier, you've dealt with this in your in your adventures. You do the same experiment twice, you don't get the same result, which is, I guess, science. But the problem is when there's a systematic effect, when lab one has a systematic difference from lab two, then when you cluster them,
Just, I was going to say, the same experiment with a deterministic, system and like, of computers, and sometimes you don't get the same results. So like, yeah, with cells is like all over the place, I can imagine.
absolutely. And if it is random variation, like it was sort of just this white noise kind of thing, well, you can average it out and you can say whatever, there's noise. But if it's a systematic batch effect, a technical artifact that is predictive of the lab in which you did that experiment, then that is a problem, because now you don't know whether you're measuring cell state or whether you're measuring the fact that it was done at the Broad Institute on day 5, right? And that batch effect problem has been pervasive in all data types, but I think it's been most obvious in high throughput, high dimensional, information rich assays like cell painting, like transcriptomics, where you're able to just take that as an input and predict the output, and you're able to get it. And it's so depressing when you're able to predict the well in which you image something, as well as you can predict which chemical you
Some kind of
do.
adverse training for it or whatever, right?
Absolutely. So there are many methods.
Yeah, I was going to say, like, it's like, one of the things that, I mean, maybe a lot of people have heard of is like when folks try to use image recognition algorithms on X-rays, right? And then one of the first things that the algorithms picked up was like, the notes, at the bottom that indicate which hospital, like that X-ray was taken or whatever. So like, you try to predict what is the result of this. Well, the X-rays that were taken at hospitals in more affluent areas had better results than the, hospitals that were, in like less affluent areas, for example, right? So the algorithm was basically predicting, was this taking, at the Mayo Clinic, or was this taking out of, some, random hospital in the middle of, Africa, right? And then, like that was, kind of one of those things that, as far as I understand, it hasn't even been resolved yet, right? Like it's maybe part of the reason why we haven't, applied a lot of these methods, routinely.
Absolutely, yes, and I think especially when you're dealing with relatively, actually, in some ways, data poor situations where, like, let's say you just have one lab that has run 50 compounds, like it's very hard to then deconvolute the batch effect from the actual signal. So there are labs that are doing. We created a baseline and an evaluation framework for this, published last year. John Aralo, a former postdoc in the group now at Genentech, who's now led that work to show that you can to sort of create this framework in which you can evaluate different batch correction methods. And our conclusion from that was none of the current methods work well enough. Some of them work a little better than others. There's one called Harmony that seems to outperform others, but it's still not a solved problem. So I would say it's a fundamental problem in the field. I think it needs a combination of better ways of tracking metadata and all that, but then also some innovation on the fundamental methods that we are thinking about. There are many others, but I know we have limited time, and happy to take other questions.
No, yeah, I mean, I was would say, one of the things that is very exciting to me, but I think maybe it's maybe the world is not ready for yet, is like robotic automation experiments, right? Like, I think, I mean, you see this a lot more. I mean, I don't know if it's more or less, right? But like when I talk with people that do experiments on animals, for example, right? So much it depends on, like, the technician that is doing the experiment. Some people are better than others. Some people, see things differently. Some people handle the animal differently, right? So a lot of it varies depending on the actual person that is doing it. Now, if you could automate it so you have a robot doing it, the robot is just running a program, right? Like, so you would run the exact same thing. It doesn't matter where the robot is physically located, in the United States or in China or anywhere else, right? And I think in chemistry, there's quite a few people that are trying to automate, workflows and experiments, using robots, right? I mean, I wonder if, I guess, like, even though we're running high throughput experiments, we still have, people that do, some of these things, that design the protocols, that run the protocols or whatever. So I wonder if, fully robotic automation would be reasonable, that it would help some of these, variation problems.
Oh yeah, absolutely. I think that will be a crucial part, perhaps the most consequential part of this whole effort, which is complete, like, or near complete automation, along with metadata that is tracked for that automation. Because robots go wrong too, but when they do, at least what might have happened, right? Not the case with human operators. I do think still that the batch effect issues will remain, because then you'll have other things like which the batch of the stain or the batch of the cell lines that you use, so many different things, right? But absolutely, I think the automation aspect of it is going to be critical. I'm so jealous of rich pharma labs and startups who have the infrastructure for being able to do that. One of the reasons why companies like InSitro and Recursion and many others are able to productively produce and use so much of their data in this fashion is just because they're able to, like, heavily control the way they do their experiments, and also track every aspect of that. So yeah, critical aspect of it, I would say.
Maybe a little bit of a hot take, just because it being controversial, right? So like, recursion in situ, like they have built, really interesting technology. They've been using it for years, but I mean, as far as I know, like they don't have any approved program, right? Like if I remember right, I think recursion may have some, they have done some phase twos, if I remember right. I don't know if they have any phase three. In situ, I imagine it's a little bit, like behind, right? But just they've been doing this for a little bit, short of time, right?
Yeah.
So in spite of how intuitively it makes so much sense, right? Like, you do this, you get a lot more data, you can do a lot more things than other folks, right? But like, why hasn't that resulted in new molecules, new drugs, new drug approvals?
Yeah, yeah. I would love to know the answer myself. I don't know. I have as you correctly pointed out, the just the fact that you're able to do things in more sophisticated ways and at scale, doesn't necessarily mean that you're going to translate to medicines. And I honestly don't know. I do think that I do think that this is a necessary but not sufficient, maybe, criteria for success, which is to take the platform approach. I know that biotech recently, I mean, at least investors have sort of pushed back against the idea more recently. I'm not in that field too much. I mean, I'm not in that circle too much, so I don't know. But it seems like anytime we talk about platforms, people are a little less excited than they were before. But I do think that is a foundation for doing any discovery. I think doing things systematically at scale is needed to understand biology systematically. And that, I think, is the more sort of more predictable path to discovery. But discovery is hard, and I don't really have an answer. I'm still bullish about it, and that probably 5 years from now, if you still have no drugs, we should have a major rethink. But at least where I'm right now, I feel like this trajectory is the one that makes the most sense to me.
Yeah. Yeah, I mean, it's like one of those things that, I mean, honestly, you could say it's the same thing about many other fields, right? Like, in this, I'm a lot more familiar about computer simulations, right? You could say, well, we've known how to do simulations for a long time. And, in general, people have been trying to do rational drug design for quite a bit longer. And, it works in many cases for sure, but like nowhere near to the overwhelming competitive advantage versus the old traditional way of like, you
Mm.
know, uh, like just, picking compounds from nature that seem to do something and then, adjusting them a little bit, right?
Yeah.
So yeah, I mean, I guess, in some ways, like, I don't know, maybe we're victims of this, um, bias that, more is better, right? So more data is always better, more automation is always better and so on and so forth. And sometimes we're kind of like missing, maybe something fundamental.
I fully agree. I think it's good to be clear eyed about that, and keep some sort of a log and say that in 5 years, I believe X will happen. If X doesn't happen, you should give it a hard look.
Yeah. Maybe kind of, I'm almost running out of time, but like, I wanted to ask, one question. I was actually just reading yesterday, this really, really interesting thread about how, some AI methods are just becoming, much, much better. Like, we may get to a, AGI or ASI or whatever we want to call it in a situation in a few years. That anyway, I won't get into the details of that, right? But like maybe the interesting part is that we will have smarter AI scientists, powered by smarter language models, probably very, very soon, right? So maybe, you know, like how would you speculate that would affect, in your case? Like we may have, scientists that automatically design and run experiments, with robotics, or like we would have, scientists that kind of extract insights from the data in a way that no human would ever, have found out. Or like, how do you think, if we had this amazing tool of like a, GPT-8 or whatever, right? That is the smarter than, the smartest, human, scientist, or like, how would that change the field, or what kinds of things, would come out of it?
Yeah, yeah. I find it very hard to predict, especially the future, as someone said. Uh, but uh, but I think the field, the subfield to very closely watch for everyone is agentic coding. I think the role of of the role of the agentic approach of LLMs in speeding up software engineering is a very, I think, will unlock or will hold the answer to a lot of the other questions, including science. Science is harder than programming in some ways, but I'm seeing, I think, the same paradigms, the same technologies, and same, I don't know, agentic workflows and so on that are going to work for creating complex software will can probably be adopted at least as a first draft for a co-scientist. I know these things are going to move in parallel. But what I'm seeing right now in agentic coding, because I'm maybe using it very heavily in the lab, is that even the most advanced models right now are still very bad at, to the extent that we need for of rule following in general. So you have to have very strong guardrails. But as long as you take a spec-based approach, a spec-driven development approach, you take a test-driven development approach to it, you're able to get amazing results, keeping the user in the loop, having your philosophy encoded in these Claude.md files, these agent.md files, having a log of what of your learnings, having everything in Markdown, by the way, like that helps a lot because both the machine and the human can read it, right? Those tricks and hacks and eventually what will become principles will, I think, really change software engineering. I'm pretty optimistic that that's going to happen. And I think the same ideas, which are working very well there, because there the feedback loop is much faster because it's like a dog fooding situation, right? Developers who are using it are the ones also building the AI tools. And I think the same paradigm can just be sort of transferred over to science, because in science too, you have this idea of having a protocol, having a structure, trying to create something. The discovery process is different and harder, but there's still this idea of needing to go and, instead of searching through a code base, you need to go and search through this experiments and then come up with, some surface some analyses from it. So I don't know where the future is going, but I know that I'm pretty sure that uh, following the keeping an eye on that space is going to be crucial.
Yeah, I mean, I think it's when I was talking with Andrew, right? Like, we mentioned that morning, right? Like, we're talking about, protein folding, and how protein folding is it's a very well structured problem with like with a good measure that you can iterate quickly, you can build, good models. So I think in some ways, building agents that automate software engineering is the best possible, example where you can go and apply, language models, because, it's of course like a very, economically useful task, right? And the people that are building these things are the same people that have been doing things the manual way for many years, right? And of course, so there's a very, very deep understanding of, how they should work, right? And then the iteration cycle is incredibly quick, right? I think this is one of the things that make a difference, right? You can have a model generate code, and then you run or interpret that code, or you run tests, and you can run that immediately, right? Yeah. Whereas, in any other scientific experiment, just need to wait, hours, days, weeks, right? Even longer, right? Depending on whatever experiment you're doing. So you can just do, many orders of magnitude more iterations, that you could do in pretty much any other field, right? So yeah, I agree with you. I think like the spearhead of like, AI automation is going to come from, automated, software engineering agent.
Absolutely. I'll just add there that I think to the extent that it's influencing science. I think it is already in other ways, in the sense that I routinely use it for just sometimes brainstorming ideas. Obviously, as everyone does, researching topics, keeping in mind that you still have to remain the expert in it. Otherwise, you will never be able to detect its hallucinations. So I think, to be fair, it is already like impacting science, and I can link to an article there, talked with some folks in Stat News about it. But it is, in terms of actually doing the kind of co-scientist thing you're talking about, I think that would need more time. But I think the developments in engineering are going to be the trailblazer field.
Yeah. I mean, maybe we'll see, some fields, slowly adjust to this more than other fields, right? But yeah, I mean, I think, we're going to have AI agents, as co-workers, sooner than we realize.
I agree.
Um, Cool. I mean, we're already at the almost an hour mark. Time really flew by. Super, super interesting conversation. Maybe like one last thing. If somebody wants to know a little bit more about your work or the work of the producer, I don't know if there's like a um, layman accessible kind of website where people can check it out, or they can reach out or ask questions or whatnot. I'm happy to link any of that stuff on the podcast notes. And um yeah, I think this was super, super interesting, Phil. Thank you so much, Shanta.
Thank you. Thank you very much, Javier. And I mentioned before, I've listened to a few of your podcasts, and it's amazing how much is happening right now in the field. And folks like you are helping us keep it together in terms of just being able to track all the progress. Folks like Andrew, you just mentioned, talking about what's happening in Future House. Folks in DeepMind talking about co-scientists. Sort of very quickly you're able to catch up on the development. So you're doing the field a service, and thank you for that.
Thank you so much. Much appreciated.
Right. Thank you for having me.